all, and thank you HFSP for this opportunity. It's a great conference, and it's such diverse topics, really all exciting. It's really sort of increasing my knowledge base by like so many folds. All right, so today my talk is about exploring the microbial dark matter by electron tomography. What is microbial dark matter? So microbial dark matter is basically 99% of the microbes that we know exist, but we don't know how they look like, what all molecular machines they use, and how they survive, how they live, or what are their communities. So 30 years ago, people still believed that bacteria are like bags of enzymes. And then now we know that they have very complex cellular organizations, they have cytoskeletal elements, they have defense systems, they have very complex molecular machines, and they cause a variety of different diseases through these molecular machines. And these all knowledge comes from 1% of microbes that we can culture. So 99% we cannot culture, and we know very little about them through metagenomic studies. So imagine the amount of knowledge hiding there, amount of cell biology hiding there. So that's where we are actually excited. This 99% of microbes actually live in all sorts of places. They are present pretty much every place on the biosphere. And they are present in hydrothermal vents. They are present in volcanic rocks. They're present in pretty much Antarctic ice, pretty much everywhere. So there are definitely really exciting possibilities of understanding this dark matter, but although at the same time there are key challenges. And the challenges are they're extremely difficult to culture. So the 99%, I say that it's very difficult to culture. Even getting enriched culture is very hard. So if you cannot even culture, then of course through metagenomic studies or through high throughput sequencing, you can understand like what all genes they might have, but then what all are expressing and how exactly those are forming different complexes. You can't really understand any of that. And as I said, that they grow most of the time in exotic, extreme environments, so very difficult to replicate those kind of environments to grow them. And getting even pure culture is or enriched culture is extremely difficult. And that's why that sort of cell biology remains really enigmatic. But then there are also many opportunities. They're like fantastic opportunities because they're rich in evolutionary history and information. There's vast amount of unknown cell biology there, and of course, understanding them also will tell us about different niches, how microbiology, different microbes form different. So how they form different ecological niches and also their role in human health, and of course there is potential in biotech applications. So today what I'll talk about is primarily is this particular class of archaea or superphylum of archaea called DPANNs. So this is tree of life. Here you have prokaryotes or bacteria. Here is eukaryotes, and then this is the third domain of life or the archaeal domain of life. And by now, we know that this archaeal domain of life actually facilitated the formation of a eukaryotic domain of life. And there's this DPANN archaea and Ascar archaea. These are two big superphylums. And I know that Ascar archaea are actually precursors to eukaryotic life. And DPANN archaea has also many implications in evolution of eukaryotic cells. So just like all other microbial dark matter, growing this DPANN archaea, Ascar archaea, these are extremely difficult. To date, maybe around 10 enriched cultures are available around the world, and this is a superphylum. And you can imagine that there are so many different members, and only 10 so far has been grown to enrichment cultures. And our collaborator, Hiroki Sakai at Soka University, now he moved to Riken, is a wizard in growing these DPANN archaeal cells. So out of the 10 cultures so far enriched, he has got about 7 or 8 of them. So he's like really great at isolating or enriching these cultures. Now, what are the specifics of DPANN archaea? Are they sort of any different from any other archaea or other microbes? So it turns out that they have an extremely small genome size, 0.5 to 1 MV, and because they have such a small and limited genome size, so that they lack many of the very critical cellular genes for these cellular processes. So as a result, they have to be always attached to another cell type so that they can get resources. So they are obligate symbionts. So understanding this DPANN archaea can also give us information about evolution of symbiosis. And of course, knowing more about them has implications for the tree of life. So as I mentioned, that they are really small, so they are like just about 250 nanometer in diameter, and they are kind of at the theoretical limit of life. So they are so small that, smaller than that, it's hard to sort of even imagine that life would exist at that form. And this is just a schematic showing that these cells will have to always stay in contact with another larger cell so that they can get resources from it for their survival. And this is our human frontier science team. So Hiroyuki Sakai is the one who is sort of really great at isolating these microbes. And then we do a lot of structural cell biology. I'll come to it, what I mean by that. And then we have Tessa Quax from Groningen University. She's a fantastic archaeal virologist, so she understands how viruses infect archaeal cells and their symbiosis as well for DPANN archaea. And we have Benjamin Good from Stanford University, who has really excellent knowledge in ecology of microbial life and evolution. So we wanted to study this, microbial dark matters or DPANN archaea, but as I say, they're extremely small and they're extremely hard to culture. So if they are really hard to culture, then there is obviously no, they're genetically intractable and their genetic methods are not yet developed. So in that case, you cannot tag them to fluorescent proteins, and then fluorescent proteins might not even work because they grow in such exotic and extreme environments. So in that case, how do you study? How do you know like how they look like or how they assemble molecular machines and what's the structural basis of their symbiosis? So what we decided to do is that we started using a method called electron tomography. So that's what we primarily use. Now this is a diverse audience, so I'll quickly introduce you to electron cryotomography and the power of it. So in electron cryotomography, basically it's a 3D electron microscopy imaging modality. But in this case, we're not doing traditional fixation of cells. We're not doing dehydration of cells. This is just native live cells. So basically just grow cultures and then put them on an electron microscopy grid and then rapidly plunge freeze it so that water molecules become vitreous around these cells. And then once we have done that, then we can put this electron microscopic grids with cells inside an electron microscope and then tilt it at multiple different angles. And as we are tilting it, we are getting 2D projection images. So once we collect all these different 2D projection images, we can put them back together into a 3D volume, which is called a tomogram. And in those tomograms, we have a resolution of about 3 to 5 nanometer. So basically, we can separate, we can see the interleaflet space of membranes. So that's the kind of resolution we have got in our tomograms. So just to give you one quick example. Okay, so another thing I also wanted to mention is that if you have thick samples, for example, eukaryotic cells or really thick microbial cells, then what we can also do is that we can add tomography to this another imaging modality, which is called focused ion beam instrument. And with that, we can make a thin slice of your sample, and then that sample can be also taken for tomography, and then we can start getting high resolution information. So for example, in this case, we are looking at pathogenic E. coli interacting with epithelial cells, and you can clearly see the 2 leaflets of outer membrane of bacteria. So that's the kind of resolution we have. And we can also see how cells interact with each other, and here it will come up, it will see that the bacterial secretion system that are interacting with these epithelial cells and then translocating effector proteins so that it can cause infection. So with these methods, then we decided to study DPANN archaeal cells, and again, to sort of demonstrate further like what we can do with these particular methods. So we're basically showing bacterial secretion systems that we can see in these tomograms. In this case, this is type 4 secretion system, and we can collect many tomograms and average many particles to get structures of these molecular machines, and then use AlphaFold and existing structures of subcomplexes to get even molecular models of these kind of large protein complexes. So in this case, this is a multi-megadalton type 4 secretion system, 4 times the size of ribosome, and you can get all these structure in situ at really unprecedented resolution. So we thought that, okay, we can maybe use this particular method to understand the cell biology of DPANN archaea, and this is what our collaborators were doing. So they were already looking at these DPANN archaea and host interaction using scanning electron microscopes. So these large cells are basically host cell, and these small cells attaching to them are all DPANN archaeal cells. So we started imaging some of this using cryo-electron microscopy. So these are host cell, and the small ones attaching to this large cell are DPANN archaeal cells. And then we thought that, okay, now we can collect tomograms of this. And then you can see that this large cell is basically the host cell. And then you can see multiple of these smaller cells, which are DPANN archaeal cells. They're interacting with this large cell. The large cell has S layer, of course. And then the small cell, which is DPANN, it has also an S layer, which is the sort of outer layer. And then what we found is that, intriguingly, that between these two cell types, all these a number of nanotubes connecting these two different cell types. And there are the many of those nanotubes, and they are forming very tight contact between the host cell and the DPANN archaea. And obviously, these probably nanotubes are conduits for material transfer between the two different cell types. So if we look at a tomographic slice of this, so that's a 3D volume. So if you look at tomographic slice of this interaction, then it's very clear that this is the DPANN archaea, this is the host, and there are these tube-like structures that extend from between the two cells. And what we also found is that in the host cell, these tubes are already present. They are in their kind of prime state. And when the DPANN cells are coming in contact with the host, then these prime tubes will just elongate, and they'll extend to the DPANN and then form this conduit so that they can exchange material between the two different cells. And then you can also view the top view of these protein complexes, or the nanotubes, and this is basically the S layer of the host, and then we can see the top view of this is one of the prime tubes that we can see the top view. And again, a side view of these prime tubes, so this is the host S layer, and these tubes are there just waiting for a DPANN cell so that they can extend and start transferring material between the two cells. And this antenna-like structure, we think, could be receptors that mediate the first attachment, and then after that, basically the tubes start elongating. So basically, now that we know that there are tube-like structures and they are embedded in the host S layers, we thought we would like to resolve the structure of the S layer as well as the tubes, and then try and understand the structural basis of material exchange between the two cell types. So this is a subtomogram average of the host S layer, and you can see that the host S layer looks like an array of dome-like structures, and it's C6 symmetric, and then there's a pore at the center. And the distance between each dome is about 21 nanometer. This is an orthoplane view of this S layer, a side view, and then there's about 3.5 nanometer pore at the top of each of the domes. Now, we know the proteins that can form this S layer. So this has been already previously shown that there are two proteins, SLA-A and SLA-B, so they can form the S layer. So what I did basically, because we have an in situ structure of the S layer, so we can use AlphaFold models to model the S layer into this density map. And these are the proteins that are forming the domes. It's called SLA-A. So that sort of 6 copies of that would come and form a dome-like structure. And then these are other proteins. The purple proteins are basically SLA-B. So trimers of SLA-B would connect to this dome-like structure, and each of these SLA-B would then interact with 3 other domes. And that kind of shows the fitting of this density into this density map, and that gave us a pseudoatomic model of the S layer of this host. So you can see now overall how each of the domes look like. And then these connections are basically connecting to the host cytoplasm. So now we wanted to know how exactly each of the tubes are interacting with the S layer, because we know that tubes can be in their prime state and they can elongate when the DPANN is in contact. And then this is a subtomogram average of the tubes that are in their prime state. And you can see that this is a kind of a really extended molecular machine where this is cytoplasmic membrane, the tube elongates, and then it kind of docks at these domes. And then you have this 6 necklace with pendant-like densities that are kind of forming a scaffold so that the tube can assemble just below the dome. And this is again a sort of orthoplane slice of this, a side view, and the lumen diameter for this tube is about 4.4 nanometer. So that tells us about 130 kD proteins in their folded state can pass through that. But what is intriguing here is that the tube is about 6.7 nanometer wide. So how can it pass through this 3.5 nanometer wide pore of the S layer? So there has to be some sort of symmetry breakage or a dramatic structural rearrangement at at the dome so that the tube can really extend and connect the DPANN cell when it is extending and forming this conduit between the two cell types. So again, a movie of the tube. So this is the subtopmum average of the prime state of the tube, and this is the cytoplasmic membrane. There's a pinch-like structure, so think that that could be a gate. And then this is the S layer, so the tube goes and then docks here, and these are the scaffold proteins. So overall, it's a quite complex molecular machine that supports the formation of this nanotube. And then when the DPANN comes, somehow the gate opens and the tube extends. So we wanted to also know like the structure of these extended tubes, how exactly they're extending and going and connecting to the DPANN cell. And what is the problem was that these tubes are coming out of this host cell at many different angles, and they're very complex and sometimes also flexible. So getting a structure of these extended tubes at high resolution was pretty challenging, but we still developed a method which is a guided subtomograph averaging method, and then we managed to get a sort of lower resolution structure of this tube. This is about 15 to 20 angstrom resolution, and this is the host, and this is the DPANN cell. And you can see that that's like a tube that's sort of extending from the S layer and now connecting to the smaller cell, DPANN cell. And in the DPANN cell, they have got this secretin-like density or a barrel-shaped density right there. It's kind of like a space station. You've got a rocket that has gone to the space and then docking with the space station. So somehow this cell has already identified that there's something coming out from this cell. And then it has formed this docking station so that the tube can dock there and then connect the two cell cytoplasms so that they can exchange material. So there's probably some sort of really very elaborate signaling or cell-cell communication going on there. We are also looking at to solve the structure of the S layer of the smaller cell or the DPANN cell, but then the lattice is not perfect here, so getting a high resolution structure of the DPANN S layer was really challenging, but you can still see the top view of this S layer of DPANN and then saw that also forms a hexagonal lattice on the surface. So of course the basic question is what all molecules are exchanged between the two different cell types. So for that what we did is we did a comparative proteomic analysis, and that in the presence, so basically we looked at the proteome of host in the presence and absence of DPANN, and that gave us some hits about what all molecules are sort of overexpressed when the DPANN is there, and some ideas around what all molecules might be exchanged between the two different cell types. Now, in this particular case, the surprising thing is that the DPANN actually wants resources from the host. But then why the host is assembling a molecular machine to support the DPANNs? What does it get in the exchange? So that's a key question. I'll just come back to it quickly. So anyway, so we basically had a second DPANN host interaction system also we were imaging. And in this case, what we saw is that the tube-like complex is not everywhere. In another DPANN system, we saw there's this pyramid-like really complex molecular machines assembled by the DPANN system, and that then connects the two cell cytoplasms. But we don't know what exactly and how exactly they form these complexes. Anyway, I'll just show another quick movie to show that different DPANNs actually assemble different molecular machines so that they can connect with their host. And as I say that, and then in that case, they exchange material. But then what are these different molecular machines? How exactly they assemble and what all molecules they exchange? They are all kind of unknown at this point. So this was another such example, and I'll just skip all that. But then this is just to show you that we have only looked at two different DPANN cultures, and it's a superphylum. So the amount of complexity there is just remarkable. All right, so these are sort of some of the future questions, and we're also looking at viruses, how they interact with DPANN and host, and how they form a complex tripartite interacting system. So with that, I'd like to sort of thank my group, and these are our collaborators for the HFSP fund, and this is other funding agencies, and I'm happy to take any questions.